Now we are going to take another term into account, that is diffusion pressure deficit. Do keep in mind that this term is an obsolete term now, but for understanding purpose we study it. It has been replaced by water potential because water potential is a more accurate term. Now this diffusion pressure deficit is in the terms of pressure potential, or rather you can say it is the diffusion pressure. Okay, so this DPD is, other term for it is DPD. What we have to understand is that this diffusion pressure is the pressure that is exerted by water in its pure state. Pure state of water will have least solvents, the number of least solutes will be 0 in it. It would be completely the water. So the diffusion pressure or the pressure potential of the water would be highest. So you can make it out that in the pure state of water, if I am having 100 milliliter of pure water, the diffusion pressure that would be exerted by the pure state of water would be highest. On addition of some amount of solutes, supposedly if some solutes are added into the water solution, what would happen? That these solutes are going to bring down the diffusion pressure of the water in the solution. That is quite obvious for the obvious reasons of the change in molarity of the solution. The molarity would comprise of the solute particles as well in the place of water particles. So solvent particles would be replaced by the solute particles in the given solution for the given molarity. So what would happen as a result? That there would be a loss in the diffusion pressure, or you can say not loss won't be the exact word, there would be a decline in the diffusion pressure. So that decline which comes in a solution, okay, the deficit, the decline in the diffusion pressure which comes in a solution as compared to the pure state of water is known as diffusion pressure deficit. Now you can consider it, supposedly you are having a cell which had pure water inside it, you add some solutes into it, so it would get rid of that pressure potential that it is going to have. So understanding the DPD in terms of osmotic pressure, we have the value of the deficit that is being created inside the solution would depend on the number of solutes, and when we talk about the number of solutes, we are talking about solute potential, and when we talk about solute potential, we often consider it in terms of pressure. If we have to consider it in terms of pressure and we are considering the diffusion pressure deficit in the given cell, it is better to consider these solutes in terms of pressure, because ultimately it is the diffusion pressure deficit. So we are going to take pressures instead of potential. So the solute potential gives us osmotic pressure. Okay, the solute potential is equivalent to osmotic pressure, which in turn is equivalent to osmotic potential. So we have osmotic pressure, the diffusion pressure deficit would be equivalent to osmotic pressure, and because we are considering a cell, so we will have turgor pressure that would be subtracted from it, and it is equivalent to wall pressure, you know that very well. So DPD would be the osmotic pressure minus the turgor pressure that would be generated in the given cell, which would be acting as an osmotic system. It would have certain value of osmotic pressure thanks to the solutes present in it. That osmotic pressure, when it is acting as a number which is to subtract the turgor pressure which is being generated by that osmotic system onto the wall or equivalent to the wall pressure, that is known as the diffusion pressure deficit. In simple terms, you can say that it is the measure of thirst of a cell. Okay, supposedly the DPD is higher, the cell would be more thirsty, that means it can drink more water. As in your own case, if you are highly thirsty, you are going to drink 2 or 3 glasses of water. If you are not thirsty, you will drink half a glass of water. In the same way, if the cell is more thirsty, that means it is having a higher DPD. That means the difference between osmotic pressure and turgor pressure is higher, that means the cell is thirsty. If the turgor pressure is not in greater value as compared to the osmotic pressure. That means both are nearer to each other. Then the difference would be smaller. Consider it, if the osmotic pressure and the turgor pressure are having greater value difference, you are going to get a higher DPD. But in case the values are almost similar, the DPD would be lower. So when these both values are almost similar, the cell is less thirsty, and when there is a great difference between the two, the cell is highly thirsty, and that can be understood in the terms of DPD. That means diffusion pressure deficit, which means a cell with higher DPD is having more concentrated protoplasm inside it, while that with a lower DPD has less concentrated protoplasm inside it. So this you have to understand. Now we are going to understand it in terms of a cell, that how the movement of cell would occur, movement of solute, not solute, how the movement of solvent would occur if we have two cells. We are going to understand DPD from that point of view. Again, I would repeat, this is an obsolete term. We use water pressure, water potential term, which is more accurate. But for understanding purpose, supposedly this is one cell and this is another cell. Both of them are in contact. Now we have to measure the DPD. Now DPD would be between these two cells and whatever solution is present over here, the solution that they are in contact with. Okay. Now you know that solution, the potential of solute potential of the cell, let's consider it to be 10. Okay, and we have the solute potential which would be equivalent to osmotic potential, and there is wall potential as well, that is turgor pressure. So we have turgor pressure, that is 7. Then automatically the DPD of this cell would be 3, as you can make it out. Okay, now you can see that this cell is having DPD of 3. Similarly, if we have for this cell, supposedly it is 5, osmotic pressure is 5, and the turgor pressure would be 2, not 2, consider it 4. So as you can make it out, DPD is 1. Now this value of DPD is higher, that means there would be movement of water from this cell to this cell. Okay, this is what is to be understood in the terms of DPD between these two cells. What we have seen over here is that the diffusion pressure deficit is nothing but the value that we get by subtracting the osmotic pressure, subtracting the turgor pressure which is exerted onto the wall by the cell protoplasm from the osmotic pressure. Now the value that comes, if it is higher in numerical value that comes, if it is higher, the cell tends to absorb water. That means it is hypertonic. If the DPD is lower, the cell tends to lose its water to whichever cell the higher DPD is present in close proximity. So this is all about diffusion pressure deficit. It is, as you can see in the word, it is the deficit, that means the decline. So the diffusion pressure, decline in diffusion pressure that comes in pure water when solutes are added is known as diffusion pressure